Good evening, ladies and gentlemen. Welcome to the 2012 Leeuwenhoek Lecture. But before we begin, I'll introduce myself. I'm Jean Thomas, the Biological Secretary, and also ask you to switch off your mobile phones because the lecture is being webcast and recorded for the archives and so on. Thank you. So tonight's lecture, as I said, is the Leeuwenhoek Lecture and will be given by Brad Amos. The Leeuwenhoek Lecture was originally established to recognize excellence in the field of microbiology, but now also includes excellence in bacteriology, virology, mycology, parasitology, and the subject of this evening's lecture, microscopy. The lectureship was named after the Dutch microscopist Antony van Leeuwenhoek, and it's supported by a bequest from George Gabb. Dr. Brad Amos was awarded the 212 Leeuwenhoek Lectureship for his exceptional impact on the field of cell biology through his development of confocal microscopy. Brad Amos is an emeritus research group leader in the MRC Laboratory of Molecular Biology in Cambridge and a visiting scientist at the university, currently a visiting scientist in the University of Strathclyde. He was an undergraduate at Oxford, where he read zoology, and then he came to Cambridge to do a PhD on the contraction of Vorticella, a microscopic pond organism. In this organism, he discovered and named an abundant calcium-binding protein, which is now known as centrin, was then known as specimen, and this protein has been found in all cells. And I gather it's not clear yet exactly what it does, but it's found everywhere, so it must be doing something important. Ask him. In 1981, Brad Amos moved to the LMB in Cambridge, Laboratory of Molecular Biology, where he found that John White had just set up a prototype confocal microscope. With John White and others in Cambridge, he pioneered the optomechanical development of the biological laser scanning confocal microscope, and he demonstrated its usefulness for biomedical work with fluorescent probes. The confocal microscope was taken up by Bio-Rad and was soon in use in labs all over the world. Brad Amos was actually seconded to Bio-Rad by the MRC for the next 17 years, where he designed confocal microscopes and the production line jigs for manufacturing them. Dr. Amos's current interest is in extending the confocal microscope to cover larger volumes so that every cell in a piece of tissue or an entire mouse embryo can be recorded in 3 dimensions with subcellular detail. So, ladies and gentlemen, I'm very pleased to present Dr. Brad Amos to deliver his lecture entitled How New Science is Transforming the Optical Microscope. Not the other way around. How New Science is Transforming the Optical Microscope. Brad.
Thank you, Dame Jean. When you had your last drink, you probably left about 100 cells on the lip of the glass, or the cup, or maybe the bottle. That's okay. Those cells are designed to be shed. But to a microscopist, it's a bit of a pity, because each living cell is as complex and as dynamic as an entire city. It is a beautiful thing. You have a central archive. You have transcription into a different language of many blueprints, which are taken out to thousands of factories. These factories churn out protein products of many different kinds. Some of them have a destination label slapped on them. They are loaded onto protein locomotives, which pass along protein rails to the furthest corners of the cell. In a modern fluorescent microscope, the image that you get is remarkably like the image of a city at night as you fly in from a plane. You see moving dots of light representing individual proteins. You see tracks, and here and there a diffuse glow because there are so many molecules. This whole subject depends absolutely on the optical microscope. Now, in my talk, I'm going to, first of all, say a little about the classic microscope and the old science. And I think there are two main principles there which we have to bear in mind in considering the modern microscope. I will start the modern story with fluorescence, which was a big transforming influence on the microscope, and the confocal method. And I was asked by somebody to be sure to describe all the new methods more recent than confocal. And with a heavy heart, I have undertaken to do that. I will not succeed, but I'll do my best, just describing a few of them. And then finally, I will give you some new results from my new collaboration with physicists and a software writer and electronics engineer in the University of Strathclyde, where I have taken this project and where we have at last succeeded in getting confocal images of large objects with subcellular detail, which has not been done before. Okay, so first of all, a little bit of principle and history. I think there are two principles. One, everyone knows, the numerical aperture, that is the angle of capture of the lens, has to be high. This is how we see fine detail. The other is not so well known, though you soon discover it as soon as you try to make a microscope, and that is that the prime lens has to be very small. That's crucial. That was the discovery of Robert Hooke, which he taught to Leeuwenhoek, and I believe that that smallness was actually the key discovery in the development of microscopy in the 17th century. Just to show you what I mean, I've done an experiment. I took two small beads of glass. One, the top one, 5 millimeters in diameter, perfectly formed and ground and designed for focusing light into a single mode fiber. And the other, a very imperfect fused bead from a bottle of beads made for chemical purposes and labeled Ballottini. So this awful bead in the lower picture, which I later found has a couple of bubbles in it and is really horrible, actually gives us a better image. I think I may be able to use this pointer, but if you haven't, oh my goodness, that's too fast. Go back. Yes, if I can point just there, you can see a 2 micron spacing of a grating. You can't see that at all in the upper image. Also in the upper image, the center of the star pattern is completely blurred out, and that is a sign of bad spherical aberration. And of course, it makes sense. If you know about aberrations, they are angular errors. So this diagram showing what happens to the rays going through a bead is true, whatever the size of the bead. But if the bead is very tiny, the effect of the angle projected back into the image is proportionally tiny. So Leeuwenhoek was able to use lenses which had aberrations commensurate in size with bacteria, and that is why he was able to observe bacteria. It was not the compound microscope, it was not the precise shaping of the lens, it was simply the size. Now the other principle, I'm sure, is known, looking around some faces here of people who know this far better than I do, but it is that you have to capture the light coming off at a high angle. And Leeuwenhoek and Hooke could not possibly know this because you need to have a diffraction grating, an opaque screen with a lot of parallel slits in it, in order to see what happens to a parallel beam of light because of its wave nature. It goes through, as on the bottom left diagram, goes through such a grating and is then split into a so-called zero-order beam, which goes straight through, and there are beams coming off at the side. Now, if you don't know why that is, then the best thing to do is to get a physicist to tell you and sit down with a piece of paper and pencil and work out how the rays, how the waves interfere to do this. But Ernst Abbe, who was working in the 19th century, had access to gratings. He made them by scribing a diamond film. And he pointed out that if your objective lens does not catch the first-order beams, the input to the lens is the same as in the diagram above, where you have no specimen. And so all you see in that case is a gray outline of the grating, but you do not see the lines. You can't see the spacing. So Abbe invented a figure of merit for lenses to express their maximum possible resolving power, which involved theta, or actually the sine of theta, the angle of capture. And it was pointed out to him that you could improve things by adding an immersion fluid, which made the wavelengths smaller, made the angles smaller, and therefore improved your ability to capture this information. So Abbe's quantity is actually the bottom of the slide, n times the sine of theta. His name for that was the numerical aperture, n being the refractive index. So we can never resolve better than that unless we use some modern tricks, which I will describe. Now, in 1890, as the result of a large grant from the Prussian government, the microscope was perfected. Abbe went to the Prussian government and said, I cannot develop the microscope because all of the glass comes from Birmingham and it is not fit for purpose. I need a lot of money to set up experimental glass making. So he got this public money, and that's why we got the microscope. There's an image of a pollen grain at the top, and you can see that the 1890 microscope, the 1890 microscope would have given exactly the same image, and you can see the grain by virtue of absorption as dark. Not very sensitive. Actually, the specimen was black, and you need a lot of absorbing molecules to see absorption in a microscope because the path length is so short. So this microscope is not very sensitive, not of great interest to a chemist for that reason. Also, not very specific. You see, the fourth image along has a bit of probably my shirt, I'm not sure, but it's not clear that it's a totally different material. It's much the same as a bit of pollen grain. We can't tell. Into this came fluorescence. Fluorescence gives us the ability to detect single molecules even. With quite ordinary equipment, a modern camera and a microscope, you can see single molecules. If they are in solution, you have no hope. They're in Brownian motion. If they're in a cell membrane, say a single fluorescent lipid or something like that, you can see them wandering around, diffusing. If they're fixed to a substrate, you can see them beautifully and use the full resolution of the microscope. As well as the sensitivity, you have specificity. And it all really began to explode in 1970 or thereabouts, when it was discovered that you could have antibodies to normal proteins, not just pathological ones, and so we could see the proteins in a cell fluorescently labeled specifically with many overlapping and apparently redundant so-called cytoskeletons, and everything took off. Synthetic chemistry came in also with calcium responsive fluorescent dyes from Roger Chen. And it has continued every year with more and more nucleic acid sequence hybridization probes, and culminated in the photoproteins from the jellyfish, where we can put a gene into an organism of study. We can make it produce a fluorescent protein itself in tandem with some other protein we want to study. So then the cell makes its own stain, and obligingly, often it continues to function normally, even though, like a soul in Dante's Inferno, the working protein may be carrying with it a rather sinister fluorescent object on its shoulder. Okay, now I got involved in microscope design as a result of meeting John White, who is a developmental biologist who could understand that there was a big problem here, the out-of-focus glare that you see in this image, but was also an electronic engineer and pioneer in computing. So he had the problem, he also had the solution. And so it was a terrific thing to be able to work with him. He was setting up something called a confocal microscope, in which the light source is focused onto the specimen. The spot of light in the specimen was focused onto an aperture, and so it's a kind of double discrimination. If you're too high or too low, you are poorly illuminated, but also if you're too high or too low in the specimen region, you are poorly detected because the light from you is going to be spread into a disk instead of going through the aperture. And now I'm going to show you a rather tedious video of an experiment which I made. You will hear a horrible noise. That's part of the experiment. It doesn't mean that you're getting tinnitus, so don't worry. So if I could have the first movie now. This is a working model of a scanning optical microscope. It's not actually confocal yet, but it is capable of scanning a spot of light over a specimen and picking up signal from any part of the specimen that is reflective or shiny. The essential features are this light source at the top and this lens here. The lens focuses the light from the light source to a point just below here where I can put specimens. If there is a specimen in the beam at that place, some light will be reflected, it'll go back through the lens and up, and we have a half silvered plate here which can divert that reflected light off to the side to a light detector here. You can probably hear a rather irritating sound. That is produced in an amplifier by converting the light signal into an audible tone. And I'm doing that so that you can hear the serial signal that is generated as we scan the microscope. Now I'm going to show you where the focus is by putting this piece of paper into the beam. When I put the paper in, you're going to be able to see the focus, but you're also going to be able to hear how the signal is actually collected from quite a wide range of levels. Now, over here, mounted in this sliding arrangement, I have a specimen which models a biological specimen much more realistically because it has depth. At the top level, we have a shiny metal grill with many holes in it. At the second level, which corresponds to the level of the focus, we have metal plates in 3 groups. In the first group, narrow ones, and the second group, broad and then narrow. And if we could pick up the signal from this layer, we would get the Morse SOS signal. It should go beep, 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 beep. And then at the bottom level, we have a continuous aluminium sheet, which will reflect back all the time. So I'm going to slide this across, and this is exactly how the early confocal microscopes worked. You had stationary optics, and you moved the specimen to build up an image. I think you'll agree that the Morse signal was very hard to pick up, if at all. But now I'm going to make this confocal. I have here an opaque metal plate with a hole drilled in it, and the position of the hole is set very carefully so that light from the focus actually is refocused by the lens into the hole. And that's the essential feature of a confocal aperture. So I pop it in, and the first thing you'll notice is that the sound has gone away. The signal level has been cut down enormously. But let's slide the specimen in as before. Did you hear the Morse signal? And I try again. So by introducing the confocal aperture, we've sacrificed a lot of signal, but we have gained discrimination of depth. Okay, now this was the team in the picture taken in 1986, showing John White, who started it all, and our first pre-production model. Very unusual. We got to control and design the production model of the microscope, and it spread very rapidly because it had things like a variable aperture and a high angle scanner, which meant you could actually fit it onto any microscope. There is nothing like that now. There's the image. The bottom row of images is taken with the confocal, and you can see how what is out of focus is black, and you have this beautiful so-called optical section. And given that you can make the section, you can construct an XY section as in the top image, or by having the scan going on a line and changing the focus, you can construct a section which is XZ, which has no optical counterpart at all. So this is a fish seen longitudinal section, and the fish transversely sectioned with its body cavity and notochord. Now, I have to leave what I know and try to say something about what I haven't a clue about. I'm going to start with structured illumination. This is very important. It is the hot thing at the moment. People are buying microscopes which cost £1 million to do it. In structured illumination, you use not a uniform field nor one spot, but a pattern of fringes. And to understand how this works, I want you to imagine that you have in your microscope a specimen consisting of fluorescent lines, which are very close together and parallel. So close together, in fact, that you cannot resolve the spacing. So this will just look like a uniform field. Now, if you impose a pattern of fringes as fine as you can by sending light into the two sides of the aperture of the objective and having two beams coinciding and making fringes by interference, I think you can see that you will then perhaps get moire fringes because of the interaction of these two. I tried to show this by a diagram. Here we have a zebra, which obviously has vertical stripes, as zebras should have, and there are some donkeys also. And if I had blurred this a little bit more, then this would have been a good representation of a periodicity that was too fine to be resolved. If we superimpose our sine squared type fringes, then it's obvious which is the zebra. But zebras should not have transverse stripes. Plainly, to find out the true direction, we have to use our imposed fringes at different angles, and as a matter of fact, at least 3 different phase positions. So that means 9 pictures for a single one. But it works. This is an example from the work of colleagues in Cambridge, Fanny Gergely, Stephanie Reichelt, and there are, on the left, there is a confocal image of 2 centrioles of a cell. These are bodies at the pole of a cell, and each one is like a top hat with a green crown and a red brim. The red brim is the so-called pericentriolar material. And with structured illumination, we can see that the brim has a hole in it. It's absolutely clear. It is a flat plate with a hole in it. We can't really see that in the confocal at all. So this is very exciting. Now, some of you may be thinking, how can this work? Because in a biological specimen, you don't have regular lines, so there are no moire fringes. And to explain that, I have to go into reciprocal space. But there are several people here who live there all the time. So I'm now talking to them with the knowledge that I'm leaving a lot of you behind. If we convey our image by digitizing it and calculating the Fourier transform into reciprocal space, we will have a disk of data, the radius proportional to the numerical aperture and with an origin in the center. And the maximum resolution is expressed by the distance along the radius from the origin. If we impose a fringe pattern, then we multiply in real space, which means that in reciprocal space we convolute, so we have new origins out at the edge. And that means we can look at peaks which are in the data which are along a diameter rather than along a radius. So that gives us a factor of 2 in resolution. In reciprocal space, the calculation is straightforward. In real space, you have a completely meaningless image. Now, all of these special methods that I don't really understand introduce, nearly all of them, some kind of switch-like or nonlinear behavior, because we're always trying to take a smooth wave pattern, something rather gradual in the image, and pick out just the peaks to sharpen up the resolution. And the ideal thing for that is something which has nonlinear properties like a switch. So when the intensity reaches a certain level, it flips, and that's it. if you could make your structured illumination with a nonlinear mechanism, you would have, instead of sine-squared fringes, sharply edged bars like stripes on a deck chair. And then, by this reciprocal space argument, your resolution could be infinite. Well, it is possible to sharpen it up by using a nonlinear process called photoactivation, and the image has been pushed now to 50 nanometers resolution by this method. So a combination of diffraction optics and nonlinear photochemistry. It's pretty lengthy, however, and computationally intensive and very, very expensive at the moment. Multiphoton-induced fluorescence is another way of introducing nonlinearity. In this case, we use a beam from a laser which is prodigiously high, gigawatts per square centimeter, and then a process predicted by the nuclear physicist Maria Goppert-Meyer occurs, where two photons actually enter a fluorophore and excite fluorescence as if they were a single photon of higher energy. Now, the value of this for microscopy is that the excitation is proportional to the square of the intensity. And that means that if you're focusing the light in a cuvette, as in this lower beam, you get absorption and emission from only that spot. So you have all the discrimination of a confocal, but without the need for a pinhole. When these results came out, and the method was really suggested by Colin Shepherd in 1980, but the results didn't reach the world at large until the early 90s from Watt Webb's group in Cornell. I thought the confocal was obsolete because this just has everything going for it. You don't need a pinhole, so you can have very efficient collection. You can get a bit of extra penetration because you're using a longer wavelength, and so on. But it hasn't obsoleted the confocal because the lasers are very, very expensive, have remained so, and there is a little problem, which is that although you may have conditions where the emission is proportional to the intensity squared, the damage, that is, the generation of free radicals, the photobleaching, the cytotoxicity, goes with something like the intensity cubed. So if you just use a tiny little bit of extra intensity, when you're short of signal, you turn the laser up a little bit, you do more damage than if you were using a confocal microscope. So it's not all straightforward. Okay, rushing on. Well, on the bottom there is one of these multiphoton image series, the same specimen, the same pollen grain. And you see there isn't really much difference between that and a confocal image, maybe slightly inferior because of the use of a longer wavelength. Okay, STED. I'm running short of time, but I will try and say something about that. This works by shining a beam of light onto the specimen and then shining a beam of a different light, which turns the fluorescence off. Magic. How does this work? How do you turn light off by using light? The answer is in photophysics. If you have an electron which has been raised to a higher energy level in a fluorescent molecule, and an electromagnetic wave comes by, it will then join that electromagnetic wave in the same phase and exactly the same frequency and drop to the bottom, drop to the low energy level. I don't know why it does that. That's physics. And I speculate that the electron has all this energy, but it's sitting gloomily in its room, and the electromagnetic wave is like a carnival that goes by, and it kind of falls into step. It wants to join in, and it goes. But of course, my physicist friends tell me that it's nothing like that. So this process of making an electron give off a photon by means of an electromagnetic wave is called stimulated emission, and it is the basis of laser action. And the way it is used in microscopy was invented by Stefan Hell, who imposes this red donut-shaped beam. You can make the focus donut-shaped by putting a phase plate in front of the laser. And the process is nonlinear, because as the stimulated emission beam intensity is increased, it reaches a point where it's knocked out all of the fluorescence. It's brought all of the electrons down to the ground state, and you can adjust conditions with a rather powerful donut beam so that only a very tiny central region remains fluorescent. And that region, and this is crucial, that region has a sharp boundary because of the nonlinearity. And in theory, you can make it infinitely resolving. It seems to be about 20 nanometers, the limit, with biological specimens. You can go a bit further with weird mineralogical things, color centers in diamonds, but that's irrelevant to this lecture. It's just coming into commercial availability, but it is very difficult to implement, and it works with only a small number of dyes. Finally, you can do sparse imaging, and this is, again, in rapid growth. This is where you, instead of looking at the whole ensemble of molecules, you somehow look at only one and somehow get a nice airy disk pattern, the diffraction pattern of a single point, because the resolution, according to Abbe, is the radius, but if you get the full profile, you can determine the center much more accurately than that. But how do you isolate just one molecule when it's surrounded by others that are also wanting to fluoresce? The answer is you use the photoprotein and you photoactivate a small subset of what is there. So this is the problem, the red envelope, where you can't see the location of individual centers. This is the solution, to repeatedly image and again and again record a good airy pattern for each center, calculate where the centroid is, and then finally project all of the centroids onto an image, which is absolutely fantastic. The resolutions claimed for these images vary from 2 nanometers to about 30, and they do tend to look like electron micrograph images. It's wonderful, but it mustn't move during this long imaging process. So it's applicable to cryosections and so on. It's very difficult to apply it to living cells, but Susan Cox in King's is doing it a different way. Instead of photoactivating, she is looking at spontaneous events, the so-called blinking of the probe. And we're plainly into very, very esoteric photochemistry here. And she is using Bayesian statistics. In other words, she's saying, that brightened up. I'm going to now look for a darkening event in that very spot. And that will be significant because it is in that very spot. So in other words, it is statistics which is looking forward and backward in time. Okay, so now the project that occupies me at the moment. people came to me in the early stages of the confocal microscope and said, your microscope doesn't work. It turned out always that they were trying to look at large objects. And it was a serious problem because large objects such as mouse embryos are being used for phenotypic screening of transgenic mice to check whether human genes might be producing disease or abnormalities. So we want a microscope that can handle this. The problem I thought I understood, which was that the depth resolution of a lens is a function of sine squared of theta. So it deteriorates very badly at low NA. So the low power lenses that we were trying to use had this awful depth discrimination. The point spread function was a needle. And so I thought I could maybe use off-the-shelf photographic lenses where theta was large, the ray path looked fine, and they didn't work. Finally, when I met lens designers such as Esmond Reed, who's here, is now a key person for me, I was told that the problem is that I had disobeyed the number one rule. A lens must be small for a microscope. If you make it large, you have terrible aberration problems. So even the most expensive photographic lenses will not work as a microscope objective. And so Es Reed designed a lens, multi-element lens, with such an extraordinary degree of correction that the elements had to be positioned to better than 3 micron centration accuracy. And they had to be shaped to a standard and precision, which is not normal in optics. But luckily there is a company called ICOS, which is a spin-off from Imperial College, that can do this kind of fabrication. We have the one lens, and it works. Could I have the Neurone movie? We used it as a camera lens, first of all, just to show that it has a very detailed image. So you start with a 5mm field, and these are neurons in green, glial cells in red, nuclei in blue, from a preparation made by Yvonne Vallis in the MRC. And you started with 5mm, and then you're zooming in by a factor of 10, and you can see the exquisite detail that there is in that image, much more detail than you have in any normal camera image. In fact, I had to cheat. I had to use 2 cameras of a different type, and we spliced the things together. What we needed was a confocal system that would work with the lens, and that's where things got difficult, because when Es had designed the eyepiece for this lens, it turned out that the eye would have to be about 50 centimeters in diameter. The beam that we have to put in is 30 millimeters in diameter, so we needed mirrors like this to scan it. These mirrors are made of beryllium, and even though they're back-thinned by milling, they're still quite massive. And this picture was taken in my home workshop, and that is a genuine coffee cup. The scale is correct. I was very pessimistic about this project because we were wanting to tilt the mirror by 2.5 degrees each way from the neutral position. We wanted that to be reproducible to better than 1 part in 20,000. Otherwise, we would not be doing justice to the detail in the lens image. So that was essential. So I took my lens kit up to Strathclyde, where there are people who do such things. And this is Gail McConnell in the foreground, who is a laser physicist and who helped me to build the mirror rig and set this the lens up. And John Dempster is a hero of the microscope physiology community because he makes software and distributes it for free for doing cell physiology. He takes in his stride not only the software for such a system as this, but also the analog electronics, which he designed almost as a little side issue. You see the lens, which is the black thing in the center. It's about sort of 50 centimeters long. Then there's the scan lens, and then there is a homemade rig housing the mirrors at the top. And only 3 weeks ago, Gail sent me this image, which made my heart go pitter-pat. That is the same electron beam fabricated resolution target as I was looking at in the very first slide. The grating that is second from the left is a 1 micron grating. The full image extends far out of this lecture theatre on either side because we were scanning the whole 5 millimeters, but plainly we have the resolution that we need, so the mirrors are working. And I luckily, just before Christmas, visited a place where they are able to make extremely good mouse embryo preparations. So Gavin Chapman of the Victor Chang Cardiac Institute in Sydney sent us these beautiful 10-day-old mouse preps, and Gail immediately showed that you can do with them. Oh my goodness! Can I have the first mouse movie, the third movie? This is not showing you the high resolution, but it shows that you can do a 3D reconstruction. And what that tells us is we have very good confocal sectioning, because otherwise this 3D reconstruction would not be possible. The resolution in Z would be too low. So that is very encouraging, but the real pièce de résistance is the next movie. There's the mouse with the neural tube on the left, the somites. The developing heart is the spotty thing in the center, and you have the chambers of the brain, and you can zoom in and you see individual cells. The bright dots are individual Golgi apparatuses in those cells. So I think this is the first time anyone has seen anything like this. We were able to record the whole microanatomy and then zoom in and look at subcellular detail. And we haven't even got it fully tweaked yet because John Dempster has since made us a fast scanning mode so that we can tweak the lens and trim the spherical aberration. That's the pericardium, where the bright dots are particularly clear. So it works. I think that this... Sorry?
What's the label?
It's called the meso-lens because it's being intermediate between macro and microscope objective. Oh, sorry. Yeah, you mean the yes. It's an anti-Golgi marker called GM130, and the dye is Alexa 488 because at the moment we have only the one laser. I think that this will probably be very useful, not just for the mouse work, but for neurobiology, for many, many things where you want to record a lot and you want to, at the very least, it will reduce the unconscious bias that you have when you select high magnification areas within a large specimen. It will speed up work where people are at present moving the specimen and stitching the images together, which is extremely tedious. But it's not just speeding it up. Because our working distance is now 3 millimeters, we can go in far further than you could with a high NA objective. So it's going to make possible imaging in depth, provided the specimen can be made sufficiently transparent. Some of my most troublesome visitors in the early phase of the confocal turned out to be geologists. So I would not be surprised if this finds uses in other branches of science also. Right. Now, I should acknowledge that after the industrial salary that I was getting from Biorad ended, the MRC supported me. So I've been supported by the MRC in this work for the last 10 years. I am also getting now tremendous and enthusiastic support from Strathclyde, and my 3 principal colleagues in this phase of work are Esmond Reed, the lens designer, and Gail McConnell. Es and Gail are both here and can answer questions. John Dempster is not actually here, but there are also lots of people involved in such a project. Stephanie Reichelt did the fish imaging. Has done a lot of confocal imaging. Steve Scotcher designed the central part of the lens, a very intricate piece of complex milling to make the filter holders. ICOS I mentioned. Yvonne Vallis, the neuronal preps. Ruth Pritchard worked with me for a summer. She's now a music student. I'm not sure whether she's here or not. But Owain Millington is working rapidly with biological applications in Strathclyde. Greg Norris took the all those pollen images that you saw, and I mentioned Gavin Chapman, who supplied the mouse specimens. There I stop. That's it.
Thank you very much for a splendid lecture. Now, Brad will take questions. If you have a question, will you put your hands up? And we can have a succession of hands. There are 2 microphones, one each side of the room, and you're asked, because of the recording, not to speak until the microphone reaches you, and preferably to stand up when you're asking your question. So first question, please. People are still stunned. There's a question.
Thank you very much for your very interesting and inspirational talk. Can you say something about the intellectual property protection that you're seeking and patents and matters like that, please?
Sorry, could you repeat that question?
Patents, intellectual property protection, what's your policy on that? What's your plans for this project?
How many hours do we have? In the optical field, patents are of very limited value because it can always be argued that you are using known means, design software, to achieve a widely acknowledged end. My experience when we had strong patents was that they can easily be destroyed by a powerful and wealthy company. We, throughout our confocal development, were attacked with relentless, continual litigation until our patents actually were overturned in a court in California for absurd reasons, but that is the legal position. So I propose not to bother joining the patent circus with this invention. It really does not give any protection to the inventor. It is giving away the specification of the invention to anyone who can afford the patent document. So it is literally without value.
Having said that about the patent not giving any protection, do you plan to give it to the public domain or the public library of science, or do you plan to keep it just as it is?
Well, I think what I would like to do is to develop it and to keep it within the academic milieu. In other words, to make systems myself, starting in my garage, and to have people participating in the development and paying for the development work, so that I don't have to go to a finance source, either a research council or a venture capital company, because if you lose control, then ultimately one of these big companies will gain control. And in my experience, they do not have the resources to develop such a thing. We are really in the same position as Ernst Abbe, in that the microscope companies are not profitable enough to generate the revenue that they need to do development. That's why it's taken them such an age to bring any of these new methods to the marketplace. So we have to find a different method. None of these methods, in fact, no significant advance in microscopy in the last 50 years has been made by the manufacturers. It's always been public money doing this. And the business model where a research council gets rid of the IP as quickly as possible to a manufacturer is guaranteed to bring ruin. I have seen the suppression of my invention, of a low-cost confocal, for purely commercial reasons, even after it had been brought to the market and people had started to order it. So these companies are in grave difficulty, and they will do anything to protect their existing products. And one large company told me that they didn't want to have anything to do with the mesolens because it could not be fitted to any of their existing microscopes. So these are not all-powerful, omniscient organizations. They are desperate companies expecting to fold, as microscope companies have done throughout history. They are very nervous, and they get up to some bad practices.
Is there a microphone? Thanks. So third row, and then second.
Brian Ford, I wasn't going to step in with a question, Brad, but since everybody else seems stunned and unable to think of one, I really would like to put one to you. The point you've just alluded to, namely that innovation in science has traditionally been done by outsiders and often in a garage at home, remains as true today as it ever did. But the one thing I want to know is time capture. Is the mesolens going to allow us, in fact, to follow the development of an embryo over a period of time so that we're going to watch cell migration, cell movement, infolding, and so forth? The still photograph that you showed was wonderfully enticing, but it was foreplay, and I'm wondering how close we're getting to the full act.
I don't know whether it would be improper to reply, but at the moment, because we have such large mirrors, the high-resolution scan, 20,000 by 20,000, takes 120 seconds, is it? If you use it as the basis of a camera system, then you can speed it up a lot. And we will look at the possibility of parallel devices like the spinning disk, though probably not the spinning disk. But we wanted to work with the single-point scanner first because it gives the best confocal depth sectioning, and that's why we've concentrated. That is unfortunately going to restrict us to very much time lapse rather than real time.
Second row here. I have the same question. Oh, the same question. That's a coincidence.
Right. Could I just add, the lens is very fast when you use it as a camera lens. Paradoxically, when you run it as a high-resolution confocal, the pixel dwell time is lower than in the ordinary confocal. It's down to half a microsecond. But if you use the system with a camera, you can get fluorescence imaging with no fluorescent lamp except the fluorescent tubes in the ceiling of the lab. It is so sensitive. And I think this may be of advantage in itself. In Stephanie Reichelt's lab, there are people who are doing bioluminescence rather than fluorescence. And there, the specimen gives out its own feeble light. But we were able to, with the mesolens, to image individual cells moving around in a tumor, which I think has not been done before. So we have a very fast lens.
Oh, one last question. Yes?
Could you just comment on the accuracy of Robert Hooke's hand-drawn flea?
It is absolutely accurate. Comparing with the mesolens images, I was struck with the way that the number, the shape of every bristle, every part of the body was absolutely accurate. And I was able to do a good comparison because we were able to get a human flea from a museum in Cambridge. They are extinct, apparently, in the UK. But no, my admiration for Hooke continues to increase, not only because of that wonderfully accurate engraving, but also because he was always giving the materials and methods. He gave accounts of how to make the lenses. Unfortunately, Leeuwenhoek did not. He was secretive about that. So in that sense, Hooke was the greater scientist, although Leeuwenhoek went further than Hooke in looking at living things and was prepared to look at things that had no name, which takes quite a bit of courage. So there's something to be said for both of these people with their totally different approaches.
Okay, thank you very much. I want to thank Brad for a very unusual lecture and splendid lecture. I think something there for everybody. I don't know what, I think probably Hooke and Leeuwenhoek would have been impressed, puzzled. They would have been wondering about reciprocal space, I'm sure. Some of the rest of us were, but clearly parts of the audience thought this is the bread and butter of life, so this is fine. This is the Royal Society and anything goes here. So, Brad, thank you very much for that. Now, I have to present Brad with another one of these red boxes with a certificate. And also to give you a splendid medal. And last but not least.